Protein synthesis. Today we are going to talk about protein synthesis, which by definition means the making of proteins. So why do we need to understand the importance of proteins? Well, roughly 16% of our bodies are made up of proteins. Proteins are vital for our existence, as they play a key role in the composition and function of our bodies. For example, maintaining our immune system by building antibodies, transportation of nutrients in our bodies through hemoglobin, forming structures like collagen. Proteins are a composition of 21 amino acids with building blocks of carbon, oxygen, nitrogen, and hydrogen, plus the R chain, which will define the function of the protein. So, by the end of this video, you should be able to describe the process of protein synthesis, which include the coding of the protein in the DNA structure, transcription, and translation. So let's look at the structure of DNA.
DNA was discovered in 1953 and brought to attention by Francis Crick along with James Watson. It was known to be very useful and important because it carries biological information with instructions for making proteins within the body. DNA molecules come together to form into a double helix. The double helix consists of 2 strands. Each strand has a backbone made of phosphate group and 5 carbon sugar groups. The ladder in between the 2 strands is created by 2 complementary nitrogen base pairs, such as adenine will always pair with thymine and guanine will pair with cytosine. It is vital the base pair sequence is correct to avoid any mutations later on in the development. If a mutation was to happen, complications could develop because a mutation contributes to the genetic variations. The base pairs are then further joined together with either 2 or 3 weak hydrogen bonds. Once the DNA structure is completed, it is then ready to unzip and split in the middle to create a single strand to proceed to its next function of replication. However, another process is transcription that we will look into next.
Have you ever had to transcribe something? Maybe you took notes in class, then rewrote them neatly to help you review? As this example shows, transcription is a process in which information is rewritten. Transcription is something we do in our everyday lives, and it's also something our cells must do in a more specialized way. In biology, transcription is the process of copying our DNA sequence of a gene in the similar alphabet of RNA. Transcription is the first step in gene expression. It involves copying a gene's DNA sequence to make RNA molecule. During the process of transcription, the information stored in gene's DNA is transferred to a molecule called ribonucleic acid. There are 3 stages of transcription: initiation, elongation, and termination. Initiation is RNA polymerase bind to a sequence of DNA called the promoter, found near the beginning of a gene. Elongation is one strand of DNA. The template strand acts as a template of RNA polymerase. The polymerase builds an RNA molecule out of a complementary nucleides, making a chain that grows from 5 end to a 3 end. Termination is a sequence called terminators, signal that the RNA transcript is complete. Once they are transcribed, they cause the transcript to be released from RNA polymerase. The type of RNA that contains information for making a protein is called messenger RNA, because it carries the information from DNA out of the nucleus into the cytoplasm. The newly formed mRNA copies of the gene then serve as a blueprint for protein synthesis during the process of translation.
The next process is called translation. This is where the mRNA goes out of the nucleus and into the cytoplasm. Then it is transported to a ribosome, which is the organelle where protein synthesis occurs. So what is a ribosome? The ribosome can be described as a large complex of RNA and protein molecules. For eukaryotic ribosomes, there are about 80 proteins. A ribosome has the molar mass of around 4.2 million grams per mole, 2/3 of it being ribosomal RNA and 1/3 of it being ribosomal proteins. The messenger RNA isn't directly involved in protein synthesis. For the production of proteins in the ribosome, tRNA needs to be used. In each strand of the mRNA containing the code for the proteins, there will be 3 codons. Codons are basically a set of 3 bases. This can be either adenine, guanine, cytosine, or uracil. Each one of these codons contain the code for 1 amino acid. Inside the ribosome, the mRNA passes through 1 codon at a time. As this happens, the tRNA starts to approach the ribosome. Each codon of the tRNA will bond to the complementary codon of the mRNA that has gone through the ribosome. The tRNA has a molecule attached to it, which is an amino acid. The mRNA slides through the ribosome, each codon at a time, and bonding to the complementary codons of the tRNA. As each codon is paired, there will be peptide bonds forming between the amino acids attached to the tRNA. This process will carry on until a protein is formed in the end.
In summary, our DNA contains all genetic codes which define us, and it will manifest through the proteins in our body. While you are watching this video right now, you are making proteins. The synthesis of proteins takes 2 steps: transcription and translation. Transcription happens when a portion of DNA is unswept, so mRNA can be made from DNA like a template. Just remember, mRNA use uracil base instead thiamine based. Transcription takes the information encoded in DNA and encodes into the mRNA, which heads out of the cell's nucleus into the cytoplasm. Translation occurs in the cytoplasm, where tRNA is floating around. When mRNA reaches the ribosome, this process begins by joining the tRNA with the respective mRNA codon. The ribosome will move along the chain from the 5' end to the 3' end until finding the stop codon. As a result of this process, each tRNA will bind with the codon and release at the same time the amino acid. This amino acid will bind to the next amino acid, forming a long chain of amino acids known as a protein. The new formed proteins will go to the Golgi apparatus to be delivered at the final destination, which could be for its own use or send it outside the cell. Our body has the full capacity to create these proteins, as long as we supply them with all the essential amino acids through our diet. In other words, we eat proteins. During the digestion, we break them down to the smallest particles, which are the amino acids. So we deliver them through the bloodstream to all our cells to make proteins again. The consequence of a poor diet can lead in the long term to mutations inside the DNA, which could develop in diseases. This concludes our discussion on protein synthesis. And remember, diet is the key to a healthy life.